Hello everyone, and welcome to today's webinar called Walkaway Biological Specimen Prep for the 21st Century Lab. My name is Mark Nelson. I'm the moderator and also CEO of Microscopy Innovations. I'm excited to be hosting this session today and pleased to be able to introduce today's speaker, Tom Strader, our Vice President of Product Development here at Microscopy Innovations. Today, Tom will share with you how to basically get an extra pair of hands in your lab by automating biological specimen prep for TEM so that you can work on other projects. We'd love to hear from you with any questions you have during today's presentation. We'll be answering questions at the end of the session, and if we don't get to your question during today's webinar, we'll be sure to follow up afterward. We'd like to encourage you to share today's webinar with your colleagues via LinkedIn, Facebook, or YouTube once it's posted online. And with that, I'd like to hand things over to my colleague, Tom Strader. Welcome, Tom.
Thanks, Mark, and hello, everyone. Thank you for joining us today. During today's webinar, I'll be discussing the benefits of using the ASP-1000 Automated Specimen Processor, a benchtop robotic tissue or grid processor for research or clinical use. I'll then show you how your protocols can be easily customized, review various applications, and end by pointing out additional resources available to you. Before we jump in, the key take-home advantage I'd like for you to remember today is that the ASP-1000 is fast at processing, simple to set up and use, easy to clean up, and easy to customize for your particular needs. Fast, simple, and easy. The entire system consists of the ASP-1000 robotic processor, capsules for holding specimens during processing, and some other accessories to help streamline and simplify your biospecimen prep workflow. Among the benefits of using the ASP-1000 is walk-away processing. Once you load your specimens and set up the reagents, simply press start and walk away. This frees you for other projects and gives you usable blocks of time without having to chase timers all day. Next is the opportunity for reduced processing times. Many of our tissue processing protocols let you go from post-fix rinse to 100% resin infiltration in under 2 hours. For kidney, the ASP does it in 46 minutes. I'll show some specific data on this later on. The ASP-1000 uses precisely the right amount of reagent, no more, no less. For specimen processing, typically less than the manual processing, and consequently less to dispose of as well. Another benefit of that is the ASP-1000 quickly and effectively evacuates fumes from the noxious chemicals such as osmium tetroxide, minimizing human handling and exposure. Of course, a big benefit of automation with the ASP-1000 is reduced specimen handling. Once the specimens are loaded into the capsules, they are never touched again. And finally, a benefit of automation that most people don't think of is increased repeatability, reproducibility, and robustness. In a nutshell, ASP-1000 does the work so you don't have to. It's a benchtop lab robot used to prepare common tissue types such as kidney, liver, muscle, nerve, or cells for TEM, pre- and post-embedding Immuno Electron microscopy, and specimen prep for serial block face SEM. There are other uses, however, we'll focus only on these three today for time's sake. As you can imagine, automation simplifies and streamlines your workflow for EM specimen prep by freeing you to work on other projects or even go home on time. While time must be taken to prepare all of the reagents beforehand, once you've done that and loaded your samples, you simply press start and walk away. No more staying late to finish the specimen processing. The ASP-1000 takes your specimen from fixed rinse through resin infiltration and ready for polymerization. Let's dive into the specifics of the automated workflow. First, the specimen is inserted into the S capsule, where it remains for processing and polymerization. S stands for specimen. This is the only time it is touched. The ASP-1000 processes from 1 to 8 specimens in parallel. Multiple smaller specimens can be processed in one S capsule and then removed and polymerized in a flat mold. Typically, one specimen is loaded into each individual S capsule. All of the reagents are dispensed into reservoirs on the deck, or pre-dispensed sealed reagent plates are sometimes used to save even more time. The ASP-1000 comes preloaded with several standard protocols, which can be used as is or can be modified to suit your particular needs. After you press start, the ASP-1000 performs all of the reagent addition and rinsing steps. If your protocol requires manual intervention partway through the procedure, the ASP-1000 can be programmed to send a text message, just as you can be notified when the run is finished if desired. After polymerization, the capsule is trimmed with a razor blade and then fits right in the chuck of the microtome for sectioning. In this slide, on the left, you will see going from top to bottom, first, the S capsule brand new. In the middle, the S capsule filled with resin and polymerized, and on the bottom, trimmed with a razor blade and ready for thin sectioning. On the right side, the S capsule is secured in the chuck of the microtome, ready for sectioning. As I mentioned earlier, the ASP uses precise reagent control to effectively deliver the reagent or rinsing solution to the specimen and effectively remove it as well. The up to 3 millimeter cube specimen fits securely in the S capsule, so the reagents can be applied with considerable force and frequency without worrying about loss or damage. It's important to remember that most tissue is 60 to 75% water, and that osmification early in the process increases permeability, so diffusion into and saturation of the tissue is accelerated. In addition to freeing you from actually performing the labor-intensive and time-consuming reagent exchanges, the ASP-1000 also significantly reduces the time required for processing samples. We've processed a number of different biological tissues, including kidney, liver, muscle, and cells embedded in low melting point agar. How can this be done so quickly? High-speed pumps and valving are used to exchange the fluid surrounding the specimen over 100 times per minute. Using kidney as our example, surveys of our customers have indicated that typical manual protocols take from 6 to 12 hours, basically a full workday. The ASP-1000, however, reduces this time to 46 minutes, enabling you to easily run multiple batches in a single day. So far, I've discussed how the ASP-1000 frees you to work on other projects, speeds processing, and reduces reagent consumption. I've also touched on how this system enables one-touch specimen handling, simultaneously providing the added benefit of an intact chain of custody. The ASP-1000 also reduces the training required for new lab technicians and eliminates technician-to-technician pipetting variability, thus ensuring repeatable, reproducible, and robust performance. Let's take a look at protocol customization. The ASP-1000 software is custom written for Electron microscopy specimen prep. It comes preloaded with protocols for kidney, liver, muscle, and cells, all of which are easy to customize for your lab's methods. Programming is straightforward and easy. Remote monitoring and text message alerts can be used to prompt manual interventions and or keep track of progress while you're away from the system. The ASP software offers two-tiered access, developer and operator. It's designed for an open access environment with skills-based logins. The developer writes protocols, validates protocols, and releases protocols to specific operators. Operators can log in and run existing protocols, but cannot modify anything, only stop and restart if needed. Programming the ASP-1000 is simple via drop-down menu of intuitive commands. Conveniences such as text message alerts can be set up to alert the human when intervention is required. Special features to ease methods development, such as running abbreviated protocols for verification, are included. An especially handy feature of the software is the ability to automatically create a tray map. When a specific plate and row is identified in the program, the software automatically generates a printable tray map that can be used to fill the reagent reservoirs. Since the protocol itself generates the reagent tray map, there's literally no way to get it wrong, other than dispensing the wrong reagent into the reservoir. Developers can set up each person who will be operating the ASP-1000 and include their phone numbers and email addresses for receiving notifications from the instrument. In this case, we have John, Paul, George, and Ringo, 4 randomly chosen names. The developer specifies which methods are available to which operators. As you can see from this example, John is working on immunoelectron microscopy projects, so only needs access to those protocols. Paul works over at the hospital in the pathology department where they prepare kidney biopsies. George works for a principal investigator on campus who does research on mitochondria, and they image rodent muscle primarily. Ringo works nights and weekends, so needs to have access to all of the protocols in case of an emergency. When you log in as operator, only the specific protocols defined by developer show up on your menu. Operator cannot edit or make changes to the protocols by the developer on the ASP. Above all, the operator can do no harm. In this case, Paul has logged in to run his kidney biopsy samples. The only method available to Paul is the one he needs. Another handy feature is the dialog pop-up. Here, a developer can give operator specific instructions of what to do for a manual intervention. Once the manual step is completed, the operator then presses OK, and the ASP-1000 proceeds to the next step of the pre-programmed protocol. As I mentioned before, the ASP-1000 can be programmed to send a text message to the operator. In this case, Ringo is concerned about leaving his osmium on the deck any longer than is absolutely necessary. So he gets an alert when the osmium step is complete to move it to the waste container. Now let's talk about some application. Kidney tissue is processed often in clinical pathology laboratories to evaluate renal function and diagnose disease. On the ASP-1000, it takes less than an hour to process 8 samples. The purple table here shows reagent plate map, and the blue table shows the step-by-step detail and timing of the protocol. Note that the osmium step and resin infiltration steps take up the largest chunks of time. Here are some images of kidney tissue processed on the ASP-1000 with beautiful ultrastructure. Wispy-thin kidney biopsy specimens commonly found in pathology laboratories require special care because of their small size and the need for specific orientation. We've developed a method using low melting point agar to stabilize the small specimens in the capsules, which prevents loss and ensures proper orientation. As I briefly touched on earlier in this presentation, we evaluated the ASP-1000 for repeatability. As part of this liver study, we tested to see if there is any meaningful variation from specimen to specimen over a relatively large data set. In this experiment, we did 3 runs of 8 specimens each for a total of 24 data points. As you can see from the collage of 24 images on the right, there is excellent specimen to specimen repeatability and consistency from batch to batch. This slide shows an example of rat gastrocnemus, which was processed on the ASP-1000 in about 2¼ hours. Note the beautiful, well-defined sarcomeres bounded by the Z discs in both images. Here are some more examples of excellent images from the ASP-1000. On the left, cardiac muscle processed in the same time frame as skeletal muscle, and on the right, some yeast cells processed in about an hour and a half. The ASP-1000 also automates processing for Immuno microscopy, which is a long and tedious process with many opportunities for error. Scientists at the University of Maryland, Baltimore have developed methods on the ASP-1000 for pre- and post-embedding immunoelectron microscopy. Dr. Ruching Shaw, pictured on the right, has pioneered optimizing the ASP-1000 for both pre- and post-embedding immunoelectron microscopy. Melania McLean and scientists at the Stowers Institute in Kansas City, Missouri, are using the ASP-1000 to prepare samples for serial block face SEM. This application is particularly amenable to automation since it's typically 5 days long and uses many extremely hazardous and noxious chemicals. In this case, they reduced the time from 5 days of manual processing to 2 days using the ASP-1000. Scientists at Oregon Health and Science University in Portland and Renovo Neural in Cleveland have recently replicated these results. Scientists at UW-Madison have also replicated these same results using the volume scope from Thermo Fisher as illustrated in this poster from M&M 2019. And finally, we have many resources available on our website. As Mark pointed out at the beginning of this webinar, I'll briefly touch on a few of these, which you can find there under the resources menu. We have a series of applications notes available on processing kidney, liver, muscle, and cells for standard TEM imaging. And we'll be releasing some new ones in the future on sample prep for nerve, Immunogold labeling, and serial block face SEM. We additionally have several scientific posters presented by our users, as well as scientists here at Microscopy Innovations that are available for download. For example, researchers at Stowers Institute utilize the ASP-1000's parallel processing ability to run 16 identical specimens at the same time with different reagents for each. This enabled quick and easy decision-making in the protocol development compared to running the 16 experiments sequentially. Also, Several researchers who've used the ASP-1000 have published in peer-reviewed journals. Here's an example of a publication from UW-Madison researcher Laura Tetri on postnatal hyperoxia, aimed at improving the survival rate of premature babies. We've discussed how the ASP-1000 frees you to work on other projects, speeds processing, and reduces reagent consumption. In addition, the secure chain of custody created by having one-touch sample processing virtually eliminates the opportunity for sample mix-up. Top it off with the increased repeatability, reproducibility, and robustness provided by automation in general, and the choice is easy. So finally, to reiterate, the ASP-1000 is a benchtop laboratory instrument that provides walk-away convenience and rapid parallel processing of batches of multiple specimens per run. The innovative reagent delivery system conserves reagent to contain purchase and disposal cost, plus easy setup and cleanup reduce overall effort expended even more. The software is flexible and easy to use with protocols that can be quickly customized and shared with other ASP-1000 operators. And to satisfy the ever-increasing need for safety, the ASP-1000 keeps noxious fumes away from the technician with its integrated fume removal. All of this in a compact footprint. Thank you for attending our webinar today. If you have any questions following this webinar, feel free to email me directly at tom.strader@microscopyinnovations.com. It looks like we have a few questions that came in during the webinar that I can answer now. Question 1. Do we need the fume enclosure if we have room to set this in our hood? That's a very good question. Thank you. The fume enclosure comes included with the system. However, it can be removed or actually not installed in the first place in the case of a brand new unit and put inside a hood. It takes up about the same amount of room as a Leica EMTP. Okay, question 2. How do you process cells? Won't they drain through the grid at the bottom of the capsule? Another excellent question. The grid at the bottom of the capsule has holes in it that are about 300 microns, so anything smaller than that will go through. In the case of cells, we embed them in low melting point agar to make what we refer to as a tissue construct, which is the 2 to 3 millimeter cubed size. We put that in the S capsule and process that similar to a piece of kidney or nerve or liver tissue. Okay, we're going to wrap up now, but I'll be sure to follow up with those of you whose questions I didn't answer. Thanks again for attending, and have a great day.